Welcome to the deep dive. Today, we're tackling, well, a pretty huge challenge. How do we actually map life, biodiversity in some of our planet's biggest, most stressed ecosystems?
Yeah, it's a critical question. We've got this stack of sources, but we're focusing on one fascinating new research paper.
Exactly. We're plunging into this paper that aims to give us a high-resolution view of biodiversity, specifically in crucial freshwater environments.
And it's urgent. Biodiversity loss in big river systems is, frankly, alarming. Think about pressures like pollution, all the dams going up, canalization. It's relentless.
And conservation needs good data. Right. Reliable, detailed data on where species actually are.
Precisely. But getting that kind of information for vast, often murky rivers, it's incredibly difficult, almost impossible, with the old traditional survey methods.
So our mission for this deep dive is to explore if new tech can help us leapfrog those limitations. We're looking at the paper, combining environmental DNA with remote sensing variables to map fish species distributions along a large river. The lead author is Zong. And the core idea, it seems almost deceptively simple, can we take genetic clues from the water itself, that's the environmental DNA, or eDNA,
Right, tiny traces left behind.
and merge that with data pouring down from satellites using machine learning to get these rapid, really precise maps of where the fish are.
That's the goal. And what's really kind of revolutionary or potentially revolutionary is linking these different scales. You've got the genomics, high res, sure, but it's just from one point, maybe a liter of water.
A single sample?
Yeah, snapshot. And then you've got the satellites giving you this continuous, huge spatial picture covering hundreds, thousands of square kilometers.
Okay.
The aha moment, if you like, is proving you can actually connect that single genetic point to the vast habitat picture from space. If they can do that, it unlocks these species distribution models, SDMs, for rivers everywhere.
All right, let's unpack this. Where did they actually do this work?
The study site was the Rhone River. They looked at its entire length, starting way up in the Swiss Alps and following it all the way down to the Mediterranean coast in France.
Wow, okay, so that covers a huge range of conditions. Different climates, different levels of human impact, different landscapes.
It's a perfect natural laboratory, really. You get this strong gradient in temperature, pollution levels. It means the fish communities change dramatically along the river's course. A great place to test this idea.
So let's break down the components. First up, eDNA. For anyone maybe not familiar, just quickly, what are we talking about?
Sure. It's essentially genetic material shed by organisms into their environment, like skin cells, scales, feces, mucus, tiny bits floating in the water.
And they use something called eDNA metabarcoding. That term comes up a lot. What does that specific process involve?
Metabarcoding, that's the key.
Yeah.
Instead of looking for the DNA of just one target fish species, like you might traditionally.
Like looking for trout DNA specifically.
Exactly. Metabarcoding lets you sequence all the different fish DNA fragments present in that single water sample all at once. It's efficient, high throughput. You get a picture of the whole fish community at that spot.
Okay. And how did they deploy that on their own? How many samples?
They were pretty systematic. They collected samples from 110 sites spread along the whole river.
110 spots.
Yep. And that gave them presence or absence data, basically, which of 29 fish species were detected at each site. That's your ground truth, your biological data.
Got it. So you have 110 points where you know which fish were detected. Now, the challenge is linking those points to the wider environment.
Exactly. Which brings us to the other half, the remote sensing, the satellites.
They used Sentinel-2 and Landsat-8 images, right?
Correct. These satellites give us regular pictures, and at a pretty good resolution. They allow you to measure properties of the river and its surroundings everywhere, continuously, not just at those 110 sample points.
So what kind of properties did they pull from the satellite data? What were they measuring?
They were clever about this. They extracted variables that reflect the actual habitat conditions fish care about, and they grouped them into two key categories, blue and green.
Blue and green. Okay, tell me about the blue variables first. Sounds aquatic.
It is. Purely the water itself. So things like river water surface temperature, RST, really important. Then the Trophic State Index, or TSI, which is about nutrient levels, how productive the water is.
Okay.
Secchi depth, SD, which tells you about water clarity, how far light penetrates. And finally, chlorophylla, COLA, indicating how much algae, phytoplankton, is in the water. It's all about the water quality.
Makes sense. And the green variables, what did they cover?
Green is about the surrounding landscape, the riverbanks, the riparian zone. So NDVI, Normalized Difference Vegetation Index, how green and healthy the bankside vegetation is. GPP, gross primary productivity related to plant growth,
Uh-huh.
also forest canopy height nearby, the slope of the terrain around the river, and something called a Human Modification Index, HUMODI, which quantifies how much humans have altered the landscape.
Okay, I get the blue variables, temperature, clarity, nutrients. Those seem obviously important for fish. But why the green ones? Why does, say, forest canopy height or terrain slope matter to a fish in the water?
That's a great question. It gets at how interconnected river ecosystems are. The green variables influence the blue ones. Think about it. Terrain slope affects how fast the water flows, how much sediment gets washed in.
Right, steeper slopes, faster water, maybe more erosion.
Exactly. And canopy height. That affects how much sunlight hits the water, which changes the temperature. Dense forest shade means cooler water. Less shade, warmer water.
Ah, I see. So the green factors shape the physical structure and the inputs into the water.
Precisely. You need both blue and green to get that complete ecological picture, the whole context for where a fish might live.
Okay, that makes sense. But now we hit that big methodological challenge you mentioned earlier, eDNA transport. The DNA floats downstream.
Yes. This is where the real innovation in the study design comes in. If you collect a water sample at point X, the fish DNA in it didn't necessarily come from fish living right at point X. It could have drifted down from upstream.
So how do you match the eDNA signal you detect with the correct habitat patch that it came from?
That was the key question they tested. How far upstream should we look? What spatial area holds the environmental variables that best explain the DNA we found? They realized a simple circle around the sample point wouldn't work well for a flowing river.
Because the source is likely upstream, not all around.
Exactly. So they tested different distances 5, 10, 15, and 20 kilometers. But crucially, they compared two shapes for gathering the satellite data. One was the standard circular buffer around the sample site.
The default approach, maybe?
Kind of. But the other was an upstream-directed buffer. It only looked at the habitat characteristics in the river channel directly upstream from the sampling point, following the flow path.
Ah, mimicking the likely source direction of the DNA.
Precisely. It was a way to properly connect the moving DNA signal with the static mapped habitat data from the satellites.
Okay, this is where it gets really fascinating. How well did it work? Did combining mobile eDNA and static satellite maps actually produce good predictions? How did the models perform?
Remarkably well overall. The species distribution models, the SDNs they build, they successfully captured the general patterns of where different fish species were found.
Can you quantify that? They mentioned a metric, TSS.
Right, the true skill statistic, or TSS. It's a
Yeah.
common way to measure model accuracy for presence-absence data. Their overall average TSS was 0.58.
And what does 0.58 mean in practical terms? Is that good?
It's definitely good. On the TSS scale, 0 means your model is no better than guessing randomly, and 1 is a perfect prediction. In complex ecological systems like rivers, anything above 0.4 is usually considered informative and useful. So 0.58, that's pretty solid.
Okay.
It basically confirms that, yes, this combined approach is feasible, it works, you can reliably map fish distributions this way.
So the general concept is sound. Now, what about that spatial question, the buffer size and direction? Did their hypothesis about looking upstream hold up?
It absolutely did, and quite clearly. The models that used the upstream-directed buffer were significantly better predictors than the ones using the simple-centered circular buffers.
Right, so accounting for the river's flow is crucial.
Essential. You have to think directionally when dealing with eDNA and moving water.
And what about the distance upstream? 5, 10, 15, 20 kilometers? Given the Rhone is a big river, I might guess the signal travels pretty far, maybe 20K.
That's what you might intuitively think, but this was perhaps the most surprising and novel finding of the study. The 5-kilometer buffer, the small scale they tested, actually performed significantly better than the larger buffers, including the 20-kilometer one.
Only 5 kilometers. Why would that be? If the water's slowing fast, wouldn't the DNA travel much further?
It seems counterintuitive at first, doesn't it? But think about what happens to DNA in a river. It doesn't just passively drift forever. It degrades from UV light, microbes chew it up, it settles out into the sediment, it gets diluted.
Ah, okay. So the signal gets weaker and patchier the further it travels.
Exactly. What this 5-kilometer result suggests is that, for the Rhone at least, the eDNA signal you pick up mostly reflects the fish community and habitat conditions in the immediate upstream vicinity. By the time you're looking 10 or 20 kilometers upstream, the link between the DNA found here and the habitat way up there becomes too weak, too noisy.
So the most relevant habitat information is quite local, even in a big river.
That's the implication. And it's a really important practical finding for conservation. When you're using eDNA on the Rhone, you should zero in on the habitat characteristics within that immediate 5-kilometer upstream stretch. That's where the strongest connection lies.
That really refines where you'd focus monitoring or restoration efforts.
Okay, let's shift to what environmental factors were most important. Blue versus green variables. Which group mattered more for predicting where the fish were?
The blue variables, the ones describing the water itself, generally came out stronger. They had more predictive power than the green riparian variables overall.
Interesting. And which specific variables were the top drivers? Looking across all the models, the 5 most consistently important variables were RST, river water surface temperature, that was huge. Then Secchi depth water clarity. Terrain slope also popped up. TSI, the nutrient status. And GPP, gross primary productivity, from the green category, snuck in there too.
But RST temperature was number 1.
By a significant margin in most models, yes. Temperature was consistently the single most influential factor.
Why is temperature so dominant for fish?
Well, fish are ectothermic, or cold-blooded. Their body temperature, their metabolism, growth rate, ability to reproduce, it's all directly controlled by the temperature of the water they live in.
Okay.
And the Rhone, as we said, has this major temperature gradient from the cool Alps down to the warm Mediterranean region. So it makes perfect biological sense that temperature will be the primary filter determining which species can thrive where along its length.
And did the study's maps actually show this playing out? Could they see species sorting themselves along that temperature gradient?
Yes, very clearly. They gave specific examples. Take the northern pike, Esox lucius. It's a species that prefers cooler water.
Right.
And the models predicted, and the eDNA confirmed, that pike were primarily found in the upper reaches of the Rhone, where the water is colder, clearer, moderately productive, often with more vegetation cover, conditions good for their young.
And then downstream, in the warmer sections.
You see a shift. Think about the European catfish, Silurus glanus. It's a huge fish, likes warm water.
Heat lover.
Exactly.
Ugh.
Its physiological optimum is way up around 25, 27 degrees Celsius. And sure enough, the model showed it was mainly distributed in the warmer, often more turbid southern part of the Rhone, closer to the Med.
So the fact that the models, built just from eDNA detections and satellite data, could recreate these known ecological preferences, that's strong validation.
It really is. It shows the approach isn't just statistically sound, it's biologically meaningful.
So, stepping back, what are the big picture implications here? What does this study really mean?
I think it fundamentally demonstrates that we can create reliable, remotely sensed indicators for freshwater fish distribution. This fills a massive gap. We need these kinds of essential biodiversity variables to track change and guide conservation, especially for large, hard-to-monitor rivers. This study proves this rapid survey method, coupling eDNA and satellite data, is genuinely informative.
Of course, we should touch on the limitations too. No study is perfect.
True. The authors themselves point out that the sampling wasn't quite as dense in the Swiss part of the Rhone compared to the French part, so there might be some spatial bias introduced there.
And that eDNA transport issue. While they found 5 kilometers worked best here.
Exactly. That's probably the main uncertainty moving forward. That optimal 5-kilometer upstream buffer. It's likely specific to the Rhone, or at least rivers with similar flow rates, channel shapes, and maybe sediment loads.
So it might be different in the Amazon or the Nile.
Almost certainly. The ideal buffer shape and size could vary quite a bit. To make this approach truly universal and predictive, the next step is probably to integrate more detailed hydraulic models that simulate how water, and thus eDNA, actually moves and disperses in specific river sections.
But even with that caveat, the basic blueprint combining remote sensing and genomics seems ready to be applied elsewhere, right?
Oh, absolutely. The core components, global satellite imagery like Sentinel, is freely available, and eDNA techniques are becoming increasingly standardized and affordable. So yes, this method can be transferred now to other large rivers where we lack good species data. They even cite examples where eDNA has been used effectively in places like the Moroni in South America or the Yangtze in China, although perhaps not yet fully integrated with remote sensing in this way.
And the potential isn't just limited to mapping where one species is it? They suggested broader applications.
That's right. The next step isn't just mapping pike here, catfish there. You can use the same machine learning framework fed with community data from metabarcoding to map wider ecological properties.
Like what?
Things like overall species richness, how many different fish species are in this stretch, or functional diversity, are there fish fulfilling different ecological roles, like predators, bottom feeders, weed eaters, or even mapping indicators of river health based on the fish community present.
And they also mentioned adding a time dimension, moving beyond just a static map.
Yes, that's the really exciting frontier. If you repeat the eDNA sampling over time, seasonally, or year after year, and combine that with ongoing satellite monitoring.
You get a movie, not just a snapshot.
Exactly. You can build dynamic models that track how fish distributions and habitats are changing. Think about predicting the impact of climate change as waters warm up, or how a new dam might affect downstream communities, especially crucial for migratory fish. With cloud computing power, running these kinds of analyses almost continuously is becoming feasible.
It really is quite powerful. We essentially have a functional blueprint now, showing how satellite tech and environmental genomics can work together seamlessly to get these near real-time, high-res maps of life in some of the planet's most vital and most threatened ecosystems. It feels less like science fiction and more like an actionable strategy.
It absolutely is. It moves the needle on what's possible for large-scale aquatic monitoring and conservation management. And it leaves us, and you, the listener, with a compelling thought to chew on. We've seen how effectively this combination of eDNA and remote sensing can map fish a complex river system. So what other hidden but critical forms of biodiversity might we be able to unlock, monitor, and ultimately protect using these same powerful integrated tools? Think beyond fish, maybe crucial microbes, elusive invertebrates in the deep sea, even things like shifting insect populations. Where else can this eye in the sky combined with the genetic fingerprint take us?